Hello and welcome to the Illuminating Immunity Light Photobiomodulation Therapy versus COVID-19. I'm your host, Dr. Joe Doduro, and my guest today is Dr. Lou Lim. He is the founder and CEO of Vielight Incorporated, and he heads a growing team of highly qualified researchers trained in medicine, neuroscience, physics, engineering, imaging, etc., and elite researchers around the world exploring new frontiers in photobiomodulation. Currently, he's working on light on trial for the COVID-19. And so great to have you here, Doc, and have you contribute. It's been pretty exciting time in the last couple of months for you guys, hasn't it?
Yeah, it's taken us more, I would say, close to 2 months to try to get this clinical trial ready and get it submitted. So we just submitted to Health Canada a few days ago, and so we got IRB approval already. Just waiting for Health Canada to give us the green light to do a clinical trial in Ontario, Canada. So we see there is a rapid pathway to do a study for COVID-19. So we hope to get this done, but get it off the ground as quickly as possible now. So we'll see.
Oh, that's very exciting. I think you're going to, is there anything that you want to highlight or talk about before we go to your slides? Because I know we're all anxious to see that. Anything that, or go to the slide?
Yeah, this is really an opportunity for me to give reasons why, we're doing this clinical trial. The questions that I'm going to ask is, when you're not a drug company, non-pharmaceutical, that is, not only is the virus novel, your study is very novel as well. So, and now when you're talking about light therapy, there's another novel therapy to present out there. So hopefully by the end of this presentation, people have some kind of feeling about whether this is a viable proposition or not. But ultimately, no matter what we say, no matter how many presentations and explanations and bases you present up there, what really matters most is clinical evidence, right? So we want to go through this process, go through the pain. It is a randomized study, and the study will be obviously randomized controls, so it will be conducted independently by another party, and we're sponsoring it, we're providing the device. As I will explain later, this is a home use device. And what's really interesting is today is the 28th of May, and this morning, Multiradiance announced they've started a study in Brazil. They are testing photobiomodulation with is static magnetic field therapy, which I'm not familiar with, for patients who are intubated. So somewhat different from us because they're obviously in much more severe conditions. We're trying to see what kind of recovery we're going to get from confirmed cases, but the treatment is at home. So it's a home treatment that is easier to to do in view of this highly transmissive disease.
Wow.
But I hope Multiradiance succeed. I know it just helps with this whole field of photobiomodulation going forward. Just that we are looking at different set of subjects to see how they respond.
I agree. Now, the point is you can do prevention, you can do kill the virus, you can do adaptive disease mechanisms, and when they're in the critical care, you have to have a mechanism for that. And so we're going to have a talk on the sepsis part of it and how that's going to be affected. So I think when you put this, the light on the person or on the virus or on the disease, you're looking at a different mechanism, different state. So all of those should be looked at. So let's go to the slides. Light on trial for COVID-19. Go ahead, Lou.
Right. Well, thank you, Joe, for the introduction. Now, I want to talk, this discussion is really giving reasons why we're doing our clinical trial. I'm not sure if you actually recorded the earlier discussion, but I'll just carry on. And when people, when we do eventually a press release, people are going to ask questions about the mechanisms, whether, you're credible or not. 100% there are going to be skeptics out there questioning us. And our job is to try to get statistically significant evidence to decide whether this method of treatment for COVID-19 is successful or not. So that's really what we want to do. There is a lot of discussion about all hypotheses and activating of immune system and anti-inflammatory effects and so on. But ultimately, it really has to come down to someone going out there, and could be us, it could be Multi-Radiance, who started their study, just started very, very recently, to show the evidence whether this whole method of photobiomodulation is going to succeed. Now, when we talk about antiviral effects, the only method that is really used widely is the application of UVC. And UVC does have antiviral effect. It's high energy and it's far shorter wavelength. Not even effectively at UVA or UVB. So this is where we want to say that there is a difference when someone talk about light. At the moment, sanitization and antiviral, it is UVC. Now, here we are not, in our case, we're not talking about UV light because it's ionizing, it actually mutates your DNA, and that's why it's carcinogenic. But I'll talk about it a bit more. And this is the device. The interesting thing is there's a little bit of history. This device has been out in the market for the last 2 years. So we're not figuring it out, hey, there's COVID-19, I've got to do something, let's go develop a device. Actually, we were not specifically targeting for a disease. It just to help people be healthier. And one of the ways is to improve your body's function, and which may include the immune system. But generally, it's a general wellness device that is pretty much in the US exempted from regulations, being under that category. Now, the key feature here is that it is home use. It is, in this case, for this study, is a treatment. And there is the possibility of prophylaxis, and we're looking into this study too. Now, there's a picture, but I think it's better for me to just put it on and see how it works, right? So this is the device. Let me show a little bit.
Nice.
Okay, that goes on a thymus, bone marrow thymus, and delivering light to the lung as well. And this is the nasal, which I will explain more. So it's a red light nasal applicator.
Excellent.
So it's convenient. It runs on rechargeable battery, as you can see in the picture, and that's it, home use. I just want to emphasize this is not a ultraviolet light therapy, and this is really using red and near infrared light, pretty much in the field of photobiomodulation. Now, the great thing about photobiomodulation is it has a history. This chart, this cartoon here, actually goes back more than 10 years. It was used by a lot of people. explaining some general mechanisms. There are a lot more, there are more fine-tuning since then. But I thought this is a very good place to start. And notice that it is the emphasis on red, near-infrared. Here it says near-infrared light, but it, photobiomodulation covers red too. It involves the release of nitric oxide. I will talk a bit more about that. There are growth factors for recovery. When you talk about sepsis and damage to the lungs, you want to look at, possible recovery and healing. Obviously, we're here because it activates immune system activities, the lymphocytic cells particularly. And what is really interesting too, there's also anti-inflammatory signaling. While you have this antiviral action going on, elevating the immune system, there is also anti-inflammatory signaling as well. And of course, there is tissue repair involving the growth factors and these elements in photobiomodulation. Now we have one applicator on the sternum, the upper sternum, where it is also called the manubrium of the part of the sternum. But there are some, it does multiple functions there. It directs light to the bone marrow. And bone marrow is where stem cells are produced. So the stem cells come out, the progenitor T cells are produced. They migrate to the thymus, which is just below the sternum. You can see it as the green, the green
organ.
Is an organ, yep. And also the wavelength could hypothetically go deeper and irradiate the lung, where you start also, have this activate the immune system. It activates the anti-inflammatory factors to calm it down. That's where most of the damage is going to be done. Now, the nasal portion, in my opinion, is very important too. And there are two parts that does its work as far as, say, the coronavirus is concerned. One of which is, as I've shown earlier, one of the factors that get released is actually nitric oxide through the activity of the respiratory chains and so on. So nitric oxide in an earlier study has the effect of inhibiting the replication of coronavirus. That's one thing. So there is this floating mitochondria around. This knowledge is starting to hold its own now in the field of photobiomodulation. So that's all there. Now, the other thing it does is, in this process, the mechanisms of photobiomodulation, it also actually proliferates T cells. And that's been done on some study in vitro. But if you're activating on, the mitochondria, you are possibly also activating the mitochondria of these naive progenitor cells that goes around and actually convert them into T cells. And that's been done in some studies. So that's a possible mechanism as well. Now here is actually quite interesting. And Michael Hamblin of Massachusetts General, is almost everyone in photobiomodulation knows him, published this review paper in 2017, highlighting the anti-inflammatory effects of photobiomodulation. And we all know by now one of the problems with COVID-19 is the cytokine storm. So while you are having this infection, your body is proliferating this immune system, creating all these T cells and the B cells, basically the leukocytes to do this, creating the humoral antibodies, creating this anti-inflammatory effect that causes the inflammatory environment for this immune system effect to take place. But it gets into this accumulation and goes beyond what's therapeutic and get into a cytokine storm. Then it leads to this not only the damage to the organs, to the lung, for example, it leads to sepsis. It starts to shut down the organs, and it seems to go beyond the lungs, getting to the kidney, the liver, some of the parts. And people are still learning about COVID-19, and they're discovering these blood clots. And actually, it seems very systemic. They're finding problems in the peripheral areas, in the toes. So people are still trying to get to grips. And having an anti-inflammatory effect is going to be useful if you can claim that. And there are some evidence. I think up to this point, the evidence is not very strong, but it's been implicated in a number of studies. And here's another study that's quite recent in 2017, done in Brazil, on acute lung injury induced by sepsis. And I think one of the presentations coming up is probably going to refer to this. Now, the study actually drew attention to a couple of signaling molecules. One is the interleukin-10, IL-10, which is anti-inflammatory. And that is what was brought up during photobiomodulation. And the other is interferon gamma. Interferon gamma is not really anti-inflammatory, but it is a molecule that actually helps to produce the antibodies out of the lymphocytic cells and so on. It helps in the antiviral effect. But it's interesting that interleukin-10 was produced through photobiomodulation. I'll talk about that a little bit more. So it seems like a very complicated chart. It probably is. And this was a publication by the Chinese scientists during, they did a lot of research actually coming out of China during their experience with COVID-19 in China. And I thought this was a good summary kind of chart to show what's going on. Now you see the effects where the entry of the virus, the SARS coronavirus 2, which is responsible for COVID-19 on the left side, and then there's antigen presentation while the body is trying to attack the virus and control it. There's developments of the immunity at cellular level, and the humoral immunity where there's these antibodies, antigens being produced and ready to get involved in the process. But a lot of it actually leads to cytokine storm, which is, I would say, responsible for a lot of fatalities in COVID-19. Now, where does photobiomodulation come in? This is putting together some published materials. Now, I've talked about nitric oxide, which can control actually this is quite an old study, not because it wasn't showing that photobiomodulation was producing nitric oxide, but it was showing that if you can if you have nitric oxide, you are able to control the replication of the coronavirus. It was quite generic. It didn't have to be coronavirus virus 2. It is the anatomy of the virus that makes it receptive to being treated with nitric oxide. Then you have, the stem cells being produced, and they get, they go out there producing the various lymphocytic cells, and then even with epithelial cells and so on, the various cells was it will get into a process of presenting these antigens that helps to control the viral activity. There is the cellular immunity. You build up immunity, then these memory cells are there to be present, to be able to control these viruses, and then of course the humoral side of it, the environment that's able to do that, and these are able to be supported by the stem cells activity produced by photobiomodulation on the, in our case, the systemic level at the when you stimulate the bone marrow, and when you activate the various cells from it. So the stem cells, interferon gamma, and also one of the mechanisms, and this is quite fundamental in our belief system for photobiomodulation, is the NF-kappa B, which is great, for immediate effects. So you have NF-kappa B, you have the production of the, let me go back to the earlier chart here. There's NF-kappa B, the activated protein 1. For very quick effects, you need these two, and they lead to the activity of the quick response, whatever it is. The activated protein 1 leads to not just the inflammatory, the response, the immune system response. It leads to very quick gene transcription that leads to growth factors and a bunch of things. So NF-kappa B can be problematic too, because it's linked to an inflammatory environment. It leads to the release of cytokines, various signaling molecules, leads to swelling because it expands the endothelial cells to allow for the neutrophils to go in and does its work and go attack the viruses. But the inflammatory environment is there, and it becomes, NF-kappa B has been associated with autoimmune problems because it can become chronic if it is not controlled. But the interleukin-10 that's been produced, and I mentioned, is also it actually is an anti-inflammatory signaling molecule, but it also in a couple of investigations also controls NF-kappa B, and you want to be able to control NF-kappa B, right? Now there was a recent study, I don't think I've highlighted here, that was published very recently in 2019, that talks about the crosstalk between directive oxygen species, which is one of the things that's produced in photobiomodulation, leading to NF-kappa B, and also having to control it because the evidence seems to be there is some crosstalk or a feedback loop that while you're producing this inflammatory environment to have an antiviral effect, there's also the anti-inflammatory elements produced by the body to control it. I'll talk a bit more about that a bit later. Now, when you look at what photobiomodulation is able to produce, And this was going to lead us to try and do another study on the prophylactic effects, potential effects of photon biomodulation. Prophylactic studies or preventive studies are, in my opinion, very hard to do because you really have to look at a big population. It's fairly long term, and I don't know if we can afford it, but we're going to try and have a stab at it. And but here's the proposition why it may warrant a study. Indeed, I mentioned about inhibition, the proliferation of, say, T cells and lymphatic cells, and also the ability of nitric oxide produced in the photobiomodulation process to inhibit the replication of coronavirus. So that's when you put in the nose, that's where it first gets into the respiratory system, right? The upper respiratory tract, which involves the nasal area, the intranasal area, the larynx, the pharynx, before it gets down to the lower respiratory tract. Now, if it gets into the lower respiratory tract, which involves the thorax, which involves the windpipe, and then the primary bronchi, and obviously the lungs, where the alveoli are. That's where your respiration, the gas exchanges take place. Now that's where, if you can deliver near-infrared light to these areas and stimulate first the bone marrow, as I've explained, to produce the stem cell that leads to, say, the T helper cells. They get mature in the thymus, get boosted. There are some studies that have shown that the evidence seems to point that this is able to actually stimulate the thymus. And that was a study that was published a few years ago. I think it involves Russian investigators and Dr. Hamlin on the effect on prolonging lifespan. It was an animal study, but it involved the thymus. Now, so if we can do that, you can now start treating when it gets to the lower respiratory tract. So we do this study now. We've designed it. It took us quite a long time. There's a lot of consultation between the many researchers in our company with outside party consultations and and a lot of back and forth discussion. So we came up with this. Now, one of the problems with this infection is really very hard to get into a hospital environment to treat disease patients unless you have, they're too busy. At that time when we were looking at this, there was, desperation everywhere. The ICU were being filled up and no one's going to pay attention to this. And it's very easy to be non-compliant and not being able to stick to the protocol. So the good thing is we have the possibility of doing this at home. And I think it's even more meaningful to be able to do it at home because the majority of patients diagnosed with this disease are sent home to recover until they get into a difficult phase, then you get admitted into hospital. So this is great. Now we have a chance to try and compare this treatment against control. So the control is people who are going through standard of care or usual care. Whatever, in our case, in Canada, it's whatever is recommended by Health Canada as standard of care, which is in large part actually self-isolation for 14 days, right? But we're adding this treatment to standard of care and see if we can get an effect size or a significant effect. So to draw the difference. Now to get the, the statistical numbers that we want, we are going to test on 280 subjects, 140 on standard of care, 140 on standard of care plus this device, the RX. So we're going to call it RX Plus. It's going to be called different from the original because we're actually tweaking some parameters there to allow us to have better penetration. And if it's going to be approved, in our case anyway, in Canada, this is going to be what is available. Is going to be a prescribed treatment for COVID-19 and hopefully for coronavirus infection. So it'll be a different device. Now the treatment it is self-administered. It shuts off automatically. 20 minutes. We're recommending twice a day for the first 5 days, and respecting this biphasic response in photobiomodulation, meaning that a certain amount of treatment, the certain amount of dose is good. Too much dose can lead you to a plateauing and then to a negative effect. So you want to avoid that. So after 5 days, we just say cut back to once a day, just to avoid the risk, but enough which we hypothesize is enough to continue to help you to get better and to treat. Now this is over 30 days. I don't think it's mentioned in this particular part of the synopsis. So it's going to continue to follow you. If you recovered well before the 14 days, we'll continue to monitor over 30 days and see whether this continue to have a prophylactic effect. When we get down to detailed data analysis, we can compare in more detail. And we're using something called the WURSS survey, WURSS, which stands for Wisconsin Upper Respiratory Stress Survey. And is that a stress or symptom? Not quite sure. So there are 44 questions, and we're going to rate. It is ordinal scale rating from 0 to like 7. So when you recover, we look at recovery for 3 consecutive days. And the statistical method is really to see if there is a significant change from from when you're confirmed. So confirmation is for us is the start of the treatment is when you're confirmed, you get registered, sign your informed consent form, get randomized. That's where it starts, and see how fast you can recover. If you do recover, there's a time to events. So whether you recover or you get worse, you get hospitalized, that's all going to be factored in. So comparing this particular treatment with just the standard of care.
Well, the point, one question I have, Lou, is that if you're going to have, let's say in the first 5 days, they start to feel better, I'm all better, they're going to continue using the therapy for the entire month as just for statistical purposes, as for treatment purposes, right? Because they might get better like right away, right?
Yeah. So we want them to continue on this treatment for 30 days because there's some people from cases published, published cases, people have relapsed. There are, we don't know. So we'll see. The more data we have, the better. And if the group in usual care are showing that, it takes longer, not just longer, but signs of relapse, and we are showing that they're not relapsing, it's going to be factored in the statistical study. So that is, so this is a really short summary. Once we get approved by Health Canada, we will we will register with clinicaltrials.gov and you can see more detail. But I want to also talk about why this is important. I just saw a survey, actually, I think I saw it yesterday, showing that half the American population do not want to be vaccinated for various reasons. But I think it's early because I think it's just, part of the survey is saying that because it's not out there yet, and maybe when it's available, they will change their minds. And there's some portion of the population who are actually anti-vaccination. So this is an option for people who for various reasons do not want to be vaccinated. But I have to say that we are proposing this not as a standalone treatment, although standalone treatment will give us some indication, but in my opinion, is an adjunct to vaccination. Because there are studies out there showing that photobiomodulation actually enhances the effect of vaccination. So this is a good thing, whether you're a proponent for vaccination or not. And personally, I feel that vaccination is very important because go right down to the bottom. My caveat is we cannot scale it as quick as vaccination, even if it works on its own. But it'll be great as a preventive measure. It'll be great for adjunct, and maybe it's good if the evidence is presenting independently to be significantly effective. That's a good thing. Now, there's no known major side effect. People in research for photobiomodulation, the users of photobiomodulation devices are aware that generally there's no major side effect. Occasionally there's some report, but no one's really. It's never been fatal. There's some resolution. There's some outliers who feel more anxiety or stuff like that. It usually gets resolved when you scale back the treatment. And this is where I talked about the biphasic response, the overdosing for some people do take place. So far there's not unresolvable. I think being home use and self-administered, doing self-administration is useful. And we have many devices out over the past few years as a general wellness device. We do receive quite a lot of anecdotal reports, and I'll just say anecdotal. People feel better. Their lives are better. So I guess there are other benefits as well in photobiomodulation. And I don't have to promote this too much. It's pretty much out there. And I believe it's affordable. It's a one-time investment. This device, the LED is going to last you a lifetime. And we've designed it to last a very long time. If it breaks down for any reason, there is this replacement policy that's, there's this warranty and a guarantee to support it. So it's a one-time investment. Hopefully, it's affordable to a lot of people. And we hope that based on, successful evidence, regulatory approval, getting to reimbursements to get out to a lot more people. And if you're using like any other treatment, not just vaccination, if you're on medication, it's not known to have any contraindication as far as we know. So that's a lot of flexibility. But you know what? If we do are very lucky, I'll say fortunate, because I don't know. I always say it's pending a clinical trial. There's going to be demand for us, and I'm going to say, look, we're not going to jack up prices to take advantage of the situation, but we respect that scaling for production is going to be a major challenge. So I want to put it out there. Now, there are questions out there. How are we comparing with the research, the multiple research that's out there right now? For the mainly drug research. I don't know of any medical device potential other than I mentioned another photobiomodulation device by Multiradiance in Brazil, and our device. So let's do an A/B comparison with the drug research out there. The first, this is actually just came out. Actually, there's a lot of stuff going on in COVID-19 research. This is just a very recent article published in Scientific American. I got it last week, and this is quite timely, and this is quite a great summary. I highlighted in yellow where I noticed there's quite a lot of discussions out there about what drug companies are going through in the clinical trials and what they've discovered. So remdesivir is, I think, approved under emergency at the moment by the FDA to allowing for use in the hospitals. It is still, when this was published in clinical trials, I think it is now being used. I think the clinical trials are still going on to get more evidence, but it is to disrupt RNA synthesis. Okay, I will get into this about the photobiomodulation perspective later. And then the second group is to prevent the entry into cells. Now the coronavirus, the SARS coronavirus 2 gets in like any other coronavirus gets in when the spike protein couples with the ACE2 receptor, which is an enzyme, is where it gets past the envelope into the cells, it replicates, and it goes up in multiple copies. Regeneron has been out there. I think that was probably less than 2 weeks ago, showing some early evidence of it being able to do this. And the next couple of days, on closer examination, it didn't show to be significant. So I guess they're going through the process of discovering how significant the changes are in the clinical studies, early clinical evidence. Then there is the treatment of the cytokine storm, the hyperimmune response, and also acute respiratory distress in the third group. So there is IL-6, interleukin-6 has been the bad boy among the interleukin group, for many years, and it's been associated with a lot of inflammatory conditions. And they are looking to be able to block the activity of interleukin-6. Now let's go on to, okay, keeping in mind there's these three groups. So when we propose the the photobiomodulation being delivered intranasally or through the sternal area, there is this mechanisms that are able to do what these drugs are doing. But I have to qualify again to say that these are mechanisms, they are related studies that warrant what we're doing, but we have to confirm. Ultimately, we're not able to prove all the mechanisms, but I think ultimately we're going to try to use these to explain, if we have successful evidence, to try and explain this successful evidence. Okay. Now the biological targets here on the first column on the left, these are common, published literature in photobiomodulation. Obviously, the respiratory chain. I've talked about the release of nitric oxide, actually inhibiting replication of coronavirus. So that's that. Then the proliferation of lymphocytic cells to be able to counter the virus at a point in time. So these are mainly, at the first stage of the infection in the nasal areas. The nasal areas is starting to show more prominence now because one of the symptoms is the loss of smell and taste. That's where you can argue that if the virus is attacking the olfactory area. So it's going to be important. And there is arguments to show that if you have loss of sense or loss of taste, sense of smell and taste, you have a pretty good chance that you are having actually a coronavirus or COVID-19 infection. So the nasal area is important. That's a target, in the early stage. Now, there are published studies to show that the action of light in vitro in a test tube actually do a number of interesting things. Of course, they do this gene transcription. They actually proliferate T cells, certain type of T cells, and we don't necessarily relate that to what's happening inside the body, but it is a good indication of what's going on. There was a study that was published, I think must be at least 10 years already, by a Chinese researcher, showing that it actually destabilized the envelope of the coronavirus. So it allows, it weakens the coronavirus when it attempts to enter the cells. Of course, the support of the lymphocytic activity will have to attack it. There was actually a study that was published last year by the United States, a division of the Homeland Security look that researching into a biological-based attacks, I think, that particular group, and found that when they still stimulate a virus in an aerosol environment, in contain and simulate full natural light, it's actually antiviral. It's able to contain viruses. So that's actually quite interesting. For us, we're looking at red to near infrared, and that earliest, the study I mentioned that destabilized the coronavirus, look at pretty much full spectrum light as well. So light actually does this to, an envelope weakens the virus, and that is full effect. So you can argue, we don't know which particular light. There is some research going on that has looked at actually blue light, I believe. And if it's not just ultraviolet, it's actually blue. And you pulse it, you super pulse it, it has an antiviral effect, but that's being challenged. I think it was challenged by another study. So that's some research going on that shows that full spectrum light has an effect, but we try and narrow down into what kind of light. Of course, UVC is one, and it could be the light actually destabilize the membrane of the virus. Then the other one is, then it goes down to gene transcription and photobiomodulation, giving the anti-inflammatory cytokines, and I've mentioned that already. The crosstalk of all the feedback between NF-ROS, NF-kappa B, and and what's going on in the cells that help to prevent the hyperimmune response. So then anti-inflammatory factors come into play. So I've talked pretty much about treatment and so on. And so these studies, including the drug studies, have not looked into cellular recovery. I just want to touch to say that one of the mechanisms, the fundamental mechanisms of photobiomodulation. Don't have to go through the higher level and just look at the last row. So all these, the process leading to activated protein 1, actually one of the in the process leads to growth factors. And I mentioned before, growth factors involve wound healing and cellular recovery. So it could be a healing of your lung after the infection. It may accelerate it. So this is a hypothesis that your 30-day clinical trial may be able to show in the data. We don't know. We'll just search out and see. And so Dr. Michael Hamlin and I wrote a paper that we did not publish because it's meant to be a white paper to support this clinical trial, asking the question, at that time it was the X Plus that was existing, whether it can be a therapeutic intervention for COVID-19 infection. So what I've said so far up to this stage is pretty much laid out in detail. In this paper, the link is at the bottom of the slide. So go there and you'll find pretty much a detailed paper.
Will we have this paper, this PDF, the white paper, included in the summit notes?
Yeah, okay, great. Right, I want to thank my big team. We have a lot of researchers in our company. I say half the staff are researchers. We're a small company, but we collaborate with a number of universities, so we leverage on a lot of skills that are out there, especially in photobiomodulation and all the various researchers. We are actually pretty much focused on the brain. And if so, Joe, you have the Brain Summit recently, and people can go get the link to our my presentation there. That's just entirely focused on the brain and what we're doing in the future. And as we expand, we're going to more facilities. So we're a small, but we're a expanding company. Our research is expanding. So I want to acknowledge that everyone in the company, and especially the last 2 months, has been pretty focused on preparing for this clinical trial for COVID-19. Right, I think, thank you. I think we're done.
I think you did a
Questions?
great job. Thank you, Lou. Thank you for your contribution. Thank you for the education, the summary that you did. That was hours of work and decades of research. So thank you so much. And we look forward to the next couple of months as you keep moving forward with your process. And let's see people getting better, huh?
Well, thank you. Yeah, it's fingers crossed right now. So we'll see. What I've presented is just stuff that warrants this clinical trial. So looking forward to it.
Let's get the light. Let's get a signal. Let's get light at the end of the tunnel.
Right. Thank you.